Okay, welcome back. This is our fifth segment on gene expression. Last time we talked about the genetic code and how the information in mRNA can be decoded using that code to direct the ribosome to link together amino acids in a particular order based on the nucleotide sequence in a gene in the DNA. So remember, the sequence of nucleotides in the DNA in the gene is read in sets of three. So that means there are three nucleotides forming a codon, also known as a triplet, and the genetic code decodes those triplets, those codons, and tells the ribosome basically what amino acid corresponds to those triplets. So we talked about how the genetic code is redundant, and we talked about how it was ambiguous. We're going to continue our discussion in this segment of how the ribosomes decode the information using the genetic code. And the first thing we're going to talk about there is tRNA structure. tRNAs are a critical part in the decoding process. They work with the ribosome to help the ribosome interpret the meaning of individual triplet codons. So here we've got a diagram of a tRNA molecule. So this is a transfer RNA. That T there stands for transfer RNA. So let's start off with what it transfers. So transfer RNA molecules transfer, or they move, amino acids from wherever the amino acids might be located in the cell, in the cytoplasm, to the ribosome. And then the tRNA decodes the information in the triplet and determines whether the correct amino acid to insert into the polypeptide, the growing polypeptide chain. So there are some important parts of the tRNA to look at. So let's start looking at the 3' and the 5' end. Here is the 3' end of the tRNA molecule, and that's the important end because that's where the amino acid, or an amino acid, will be attached to the tRNA. So there are 20 different amino acids, and there are a whole range of different tRNAs. And so I think there are 64 or something tRNAs, and so there's redundancy, remember, so that means that some tRNAs carry the same amino acids because of the redundancy in the genetic code. So there are a bunch of enzymes which add amino acids to this end. Each tRNA is going to carry only one amino acid, and so the amino acid gets added to this end by an enzyme in the cytoplasm, and the amino acid that's carried by the tRNA depends on this region down here called the anticodon. So the anticodon determines what amino acid will be attached up here at the 3' end of the tRNA. So the most important thing about the anticodon is that it's going to be complementary to the codon sequence. So this anticodon here, we see GGC. So that corresponds to a codon, which would be CCG, written 5' to 3'. So what happens is if this anticodon matches a codon, the ribosome knows that whatever amino acid is attached to this end is the right amino acid for that codon. So let's just recap that. If the codon in the mRNA is CCG, which is complementary to GGC, which is anticodon, the anticodon and the codon will match. They will be complementary. The C's and the G's will match up. And that means the ribosome knows that whatever amino acid is attached here at the 3' end is the right amino acid to add to the polypeptide that's growing. So the rest of the molecule, you can see there are stems, there are some loops. It's a complicated three-dimensional structure. And then over on the right here, you can see the three-dimensional nature of the tRNA molecule. RNAs aren't linear. They're linear in that they have two discrete ends, but they're highly folded, as you can see here. And then down here again is our anticodon. So the anticodon is a critical part in deciphering the genetic code. So for every codon, there is a complementary anticodon, and the anticodon of a particular tRNA tells the ribosome which amino acid is appropriate to be inserted into the growing polypeptide chain. So let's take a look at the ribosomes, and we're now going to move into actually talking about translation and how you have translation initiation. That's the beginning of translation. So two diagrams, two representations of an amino acid. Let's start with the simpler diagram on the right-hand side here. So over on the right-hand side there, we can see there are actually two subunits. So I'll highlight the two subunits so you can see them easier. I'll try and do it quite quickly, so it'll be a little bit messy. Okay, there's one subunit. That's the subunit which we can set to the bottom of the ribosome. That's the small subunit, and then there's what's called the large subunit, and that's up here. And they're often called the 30S subunit and the 50S subunit. And you don't need to worry about where those names come from. You just need to know there are two subunits. One is small and one is large. These are assembled in the nucleolus, in the nucleus. And then they're exported out of the nucleus through the nuclear pores. And it's only when translation initiates do they come together. Otherwise, they are kept separate via various mechanisms, which aren't important. And they only come together at translation initiation. So that's the overall structure. And then you can see there's an mRNA here that's going to be translated. And we'll come back to this process of translation in a moment. Then there are four important sites that we need to worry about. There's the so-called A site, a P site, which is in the middle there, and the E site, which is over on the left as we're looking at it. Now bear in mind, there's no left and right. There can also be a middle, but there's no left and right. It depends on which way around we're looking at the ribosome. And then we've got this this exit, or this little passageway here, where the growing polypeptide will leave. It'll be threaded out of the ribosome. And so those are the important structures of the ribosome. Now when we look at the two subunits, let me just outline them again. So here is the large subunit, there, kind of on the top, and then here is the small subunit. And you can see there's kind of some squiggles and some small balls in each of these subunits. So the balls there represent amino acids, so that's protein. So there's both protein and then all these gray and light blue kind of squiggly structures. Those are actually ribosomal RNA. So ribosomes are made of two macromolecules, RNA and protein working together. Now the interesting part about the ribosome is that the RNA gives the overall structure, which is then kind of fleshed out by the proteins. Possibly the most important and interesting thing about ribosomes is that they, the reactions they catalyze, the chemistry that they do, the linking together of the amino acids, that's actually done by a piece of RNA. So that's a catalytic piece of RNA known as a ribosome. We don't have to worry about that too much. It's interesting to point out that not all enzymes are proteins, that some actually are RNA molecules, and this hails back to kind of the early RNA world that you'll have heard about, you may have heard about. You've seen actually other catalytic RNAs when we talked about splicing a little bit earlier. That was also an RNA enzyme. So that's the general architecture of the ribosome. Let's take a look at the process of initiating translation, and this will help you understand a little bit more about how this decoding process occurs using the genetic code. So the first thing that's going to happen when you start translation is that an mRNA in the cytoplasm of the cell, so this always begins in the cytoplasm of the cell, the mRNA, here's our mRNA, it's going to be grabbed by a small ribosomal subunit. It's grabbed up at the 5' end, and then it scans, it being the small ribosomal subunit, it scans along until it finds the first AUG that it comes across. And that AUG is always going to be in the context of something called a ribosome binding site right here. So that's ribosome binding site here. It's not actually a site where the ribosome initially binds. It's actually a sequence of nucleotides which is complementary to one of those ribosomal RNAs. And when those two slide past one another, so when the mRNA slides past a certain sequence in the ribosomal RNA in the small subunit of the ribosome, they hydrogen bond. There's a region of complementarity, and it slows the ribosome down. It's kind of like a pausing that happens at that point. And so when that happens, the AUG is positioned in this middle site here. So remember, we've got three sites. I'll try them, write them in so you can read my terrible writing here. So we have an A site, and then in the middle is a P site. And then over on the left, as we're looking at it, is the E site. So we've got these three sites. And the AUG, remember, the start codon, is going to get positioned in the P site in the middle. So that's the kind of the first step, is to scan along the mRNA. And when this ribosome binding site gets hit, the ribosome pauses, and that locates the AUG just long enough in the P site for the next thing to happen. So you've got to imagine in the cytoplasm, you've got all of these tRNAs kind of whizzing around. There's a lot of molecular motion. And so with some frequencies, what will happen is that the initiator tRNA, and the initiator tRNA is a special RNA, this amino acid is always going to be methionine. So that's always going to be a methionine amino acid. And the initiator tRNA's anticodon is going to be UAC, and that's read from 3' to 5'. So UAC is complementary to AUG. So now we've got our first example of how we are decoding the information in the mRNA, which is just a version of the information in the DNA, using the tRNA and the genetic code. So UAC matches AUG, so the U and the A can base pair, the A and the U there, and the C and the G, so they base pair. And so that tRNA, that initiated tRNA, stably sits on that AUG sequence in the P site of the small ribosomal subunit. And because there's an enzyme which knows to add the methionine amino acid to tRNAs which have UAC, so that protein which recognizes UAC in the anticodon only ever adds the amino acid methionine to the 3' end of the tRNA up here. So that's our second step. So our first step is small ribosomal subunit scans along the mRNA, hits that ribosome binding site. That causes it to kind of pause momentarily. So the AUG is in the P site, and then one of these initiated tRNAs comes in. And bear in mind, there's lots of other tRNAs trying to get in there, but they don't match the start codon. Their anticodons don't match the start codon. So UAC locks in there, hydrogen bonds form, and that keeps that initiated tRNA in place just long enough for the next thing to happen. The next thing that happens is that the large ribosomal subunit comes and docks onto the initiated tRNA. So that's the kind of third event in translation initiation. Once the 50S ribosomal subunit, the large subunit, sees a small ribosomal subunit paused at an AUG with an initiated tRNA there, it kind of jumps on. But if there's no tRNA there, it's not going to jump on. And so that is the end of translation initiation. We've got an assembled ribosome with a tRNA with its amino acid, which is always going to be methionine. And that's all assembled in the P site of the ribosome. So we're going to stop there, and then we'll take a look at the next stage in translation, which is elongation, and we'll talk about more decoding events at that point. So we're going to wrap up there, and I'll be back with you soon to talk about translational elongation.